You are listening to Science Coast with Mallory Havens and
Chris White.
And today our guest is
Sarah Powers.
Hey, Sarah, can you tell us a little bit about yourself?
So I am part of the biology faculty at Lewis. I have a PhD in immunology, so I think that's why we're talking about some immunology stuff today.
Yeah, so today we're going to talk about the mRNA vaccines that have recently come out. So if you're unfamiliar, that's what the COVID vaccine is based on.
Was that the first mRNA vaccine?
I think it was the first approved. Is that right, Sarah? Do you know?
And in large-scale rollout.
Yes. So, and there are many more in the pipeline that they're working on things, but we invited Sarah here today to give us her expertise on how these vaccines work as an immunologist.
I'm wondering if we could start by just talking about how vaccines work generally, and then move on to how mRNA vaccines differ from traditional vaccines.
Sure.
Okay, so vaccines are fantastic because they help to prime or initiate educating your immune system to know how to respond to a pathogen. Instead of you having to encounter it for the first time with an infection and then have your cells that need to get themselves in gear to be able to respond and help fight it off. So vaccines give us a leg up and hopefully make sure that if we're exposed to the pathogen, then we could go ahead and clear it much more efficiently.
So it's like getting a head start. The body is given a head
Bingo.
start.
It is a really big head start. And it's important for cases of diseases where our immune system might not be the best at clearing it naturally, or there are parts of our population that are less efficient at clearing it so that there's higher incidences of death because you can't clear the infection, or there might be long-term consequences if you do recover, but there could be paralysis or there could be other long-term consequences of not being able to go ahead and get rid of it.
So it allows the body to ramp up and fight the infection, whatever type of infection it might be, before the infection has an opportunity to do serious damage.
Bingo.
Okay, cool. And they work.
They work.
I just want to be explicit about this.
Yes, Chris, they work.
Okay.
Have you ever had smallpox or mumps or measles or?
No, I'm not saying that I don't believe in vaccines. Okay, so how were vaccines made previously, like before this technology?
There are a few different approaches that are used in different instances depending on the thing that makes us sick. So there are some cases in which a vaccine is going to be based on what we say an attenuated or a weakened version of the actual pathogen. This happens with certain viruses. So what you will do is train the virus to be not as efficient at infecting human cells, but other kinds of cells instead, with the idea that you then give that in selected doses to the human as the vaccine to amp up and go ahead and get their immune response started. The caveat to that is that individuals with a weakened immune system really aren't good candidates, because even if it's an attenuated or weakened version of the virus and their immune system's not working, they could actually get sick from it. One of the other approaches is to go ahead and take the pathogen and heat inactivate it and then use parts of that in a vaccine in order to stimulate. The problem is when you heat inactivate a pathogen, it's not in its normal form, and so it might not elicit as strong of an immune response. The other approach is that you could go ahead and isolate out parts of the pathogen and develop that into the context of a vaccine. The challenge there is that you usually have to use some kind of cell culture in order to make those parts, and that's usually utilizing a non-human kind of cell to make it happen. So the manufacturing process is elaborate. It takes a long time to make that, and you have the potential of carryover allergic responses that could wind up happening because of the other cell culture system. So there are caveats to these different kinds of approaches.
Is it eggs that are often used?
There is a common use of eggs in many instances.
All right,
Yeah, that's why people with egg allergies can't always get every single type of vaccine.
Mm-hmm.
So I do have one more question. I think I know a little tiny bit about it, but I'd love to hear your answer. How does the body remember? So a vaccine exposes a human to a particular type of, a protein or something that the body then knows, okay, I need to prepare to attack this when it shows up. How does the body remember that you've exposed it, but then the body clears out the vaccine, I presume at some point, and then you might get infected 5 years later? How does the body remember 5 years later that you had a vaccine?
We have something called memory cells, but I'll let Sarah elaborate on that.
Yeah, they're pretty cool. So Our immune system actually works with kind of a two-pronged approach. We have some cells that are short-lived and they're really powerful in the immediate. And they are then going to work in concert with our lymphocytes. So our T cells and our B cells, they have the potential to become memory cells. So we activate a lot of these cells that are specific to the thing that makes us sick. Initially, they expand outward so that we have a lot of those cells to clear it. And then a subset of those are going to be retained long term. And so they just live in our body. Some of them are going to be making what we call antibodies all the time. So we have these, they're kind of soluble probes that are just going to float around in your blood serum that can interact with the pathogen when it comes back. So we know that the longevity of having cells that make these antibodies can last well out beyond 80 years, probably longer. So they are going to be there for a really long time.
But I still have to get my tetanus shot every 10 years.
Well, because you want to reboost, right? So the only way in order to make sure that these memory cells persist for a long time is to kind of go ahead and give them a reminder of, and get them reactivated so that you can expand the population. Because if these cells are in circulation and you go for something like a blood draw to do another diagnostic, and you wind up just by chance having some of these memory cells that are in that blood draw, well, once they're out of your body, they're gone, right? So you're usually not relying on just one copy of these. There are a number of them. And so if we go ahead and we re-challenge with a booster, you bump up those numbers a little bit.
And it's also like learning about pathogens through exposure is why children are a vulnerable population, because they've had less exposure to all sorts of diseases. So they're going to probably have to go through that primary immune response, get sick before, so they can make memory cells. And then when they encounter it again, they're fine. And for us, adult, non-elderly population, we have that immunological memory and it's working. But then as you age, you start to lose some of that immunological memory and become susceptible again.
So as we then start talking about this new technology that was invented and the Nobel Prize that was issued, I still am not clear because I was doing some reading about RNA. And
ooh, my favorite.
And then there's there's mRNA and there's tRNA and there's generic RNA.
rRNA, microRNA, long non-coding RNAs.
I decided that if I was going to ever understand this, I was going to have to go back to school. So real quick, how many classes did you guys have to take in order to really understand this stuff?
Oh, I don't know.
How many years of biological training?
Well, I'm an molecular RNA biologist, so like just being an RNA biologist is a whole subset, and then within the RNA field, people will specify for like one type or one type of processing of one type. So it's, I don't know, to grasp the whole RNA field in depth, I don't know if there's a single person that has it all.
There's like the basic understanding, right? And then there's all these nuanced pieces, and the more time there is for research to go on, the more opportunity is to understand just how much we might not have already known.
Right.
So what do people need to know to get a better understanding of why this technology is so amazing?
Well, I guess I'll take the lead on saying what mRNA is. That stands for messenger RNA. So the job of messenger RNA is to code for proteins. There are a bunch of different RNA species that don't make proteins. The RNA itself is the end point. But when you copy your DNA into RNA with the intent of making a protein, that's mRNA. So it codes for a protein. So an mRNA vaccine is the code to make a protein that's part of the pathogen that should hopefully fold into its natural form, and your immune system can recognize that. But you're avoiding some of those problems Sarah was mentioning of heat inactivating or using a different culture. You're just giving a genetic sequence essentially so your body makes part of the pathogen in the right shape so the immune system can learn about it.
And it's pretty cool because we're essentially using this vaccine to co-opt cells of the body to go ahead and make the protein that's reminiscent of the pathogen in a way that it's mimicking what would happen in a natural infection without it actually being an infectious state, right? So these cells can make, in the context of the COVID vaccine, right, it's making the spike protein that comes from the virus. And so that's displayed on the surface of our cells, right? It gives an opportunity for our immune system to get its hands on that and be able to show that and get those T cells and B cells activated so we can get a memory response. But you don't have any opportunity for the virus to go ahead and infect any other cells because there's no virus itself there.
And it should be mentioned too that when you get a virus, it goes into your cells and it takes them over. It essentially enslaves them to make viruses. So viruses in and of themselves cannot reproduce on their own. They're essentially genetic information and protein. Sometimes they have a little coat on top of them and they might express viral proteins on the outside of that. But in and of themselves, they can't do anything. They have to enter our cells, take over our systems that are already there and use our own energy, use our own nucleotides, use our own amino acids. So essentially you're doing what the virus would do with an mRNA vaccine, but only a portion of it, so it's not infectious.
So I have so many questions because this is such a fascinating topic to me. When the vaccine, I remember when the vaccine was being developed, and there's all this talk about spike proteins, and so I'm a little bit curious about, you're not the antibodies and the vaccine doesn't replicate the entire, COVID virus. It only replicates or activates, the body to certain pieces of the virus. And then there's the potential for a virus to evolve. So in other words, if the immune system is going after a certain protein that's showing up on the exterior of the packaging, and then the virus evolves not to have that protein, it can then evade the vaccine?
It could.
In the case of what we've seen with SARS-CoV-2, though, it's not about getting rid of that protein, but it's about having minuscule little changes to its shape. And so our antibodies from our immune response are so terribly specific in their biochemistry to interact with its target that those tiny little changes in the shape are enough for the antibodies to not stick to it anymore. And so without then mounting an immune response to this new iteration of the virus-derived protein, you're kind of left a little bit in the dark.
So I feel as if I'm now ready to ask the ultimate question, which is why we're having this podcast today. When the Nobel Prize was issued to Katalin Karikó and Drew Weissman, what did they discover? What is it that they did that the Nobel Committee said, yes, that was such an amazing discovery that we're going to issue the Nobel Prize?
So we're going back to kind of the early 2000s when they were doing some of their seminal work. And they were trying to think about how we could use mRNA molecules for therapeutics in a way that when you deliver this mRNA molecule to the human body is not itself triggering an immune response because it's perceived as foreign. So it was in the heyday of immunologists and molecular geneticists and cell biologists trying to understand some of the detection mechanisms that are being used by our immune system to tell the difference between things that are us and things that are foreign. And it was a little bit perplexing in the context of things like messenger RNA because fundamentally, really all life forms use messenger RNA in this process in order to take the information that's in our genes to then make the message to ultimately make the protein like Mallory told us about before. So how in the world can we distinguish between foreign mRNA and our mRNA?
Right. Oh, no, I was just confirming.
So I think it's also worth mentioning that Dr. Karikó was the one who was really doing the mRNA research, and her colleague, Dr. Weissman, was an immunologist who was complementing, and they were collaborating, but she was the mRNA expert.
Yes, so she was doing the mRNA side, and he was really tied to providing the immune cells that they were testing this in, right, in order to see if they were getting an immunological response.
And one of the things that I thought was really interesting about the story is that the community at large, and in particular the funding agencies, 10, 15 years ago, didn't think that research into this mRNA, let me just say stuff, was worthwhile. And she had the passion and the vision to say, no, this is important work. But it did come with consequences for her. So she was pursuing this because she was passionate about it and really believed that there was, important discoveries to be made. Yet the funding agencies didn't support her. And in fact, she had, her job was, she was demoted at some point and basically told, well, your research isn't getting funded, therefore you're not faculty material. Yet here she is today with a Nobel Prize. And I wonder what that says about science in the United States.
Well, I think sometimes scientists might have a vision that isn't necessarily understood by other people in the field or other people in funding agencies. So there's always that balance between having the vision but being understood. And they kind of say about scientific funding through the federal government in the United States that you kind of have to have done the work to get the money to do the work, because they want proof of it. And they are resistant to newer and crazy ideas because it might not get the same kind of return. So there's flaws in the system for sure, but there are ways that people can still do some groundbreaking stuff that might not be commonly perceived as important through like startups and, that kind of thing.
I would add to it that I think, because ultimately these funding agencies are responsive to our taxpayers, right? And so the average American is looking for the return on this, and they're looking for a lot of things that are easily translational, so into treatments, things that can go out into studies tomorrow. And the problem is that all of those things at some point were rooted in understanding the basic science. And there's a big time delay between understanding the fundamentals to then when you can ultimately get to the point of being able to turn it into something that might help a patient in the hospital.
And that's why when people are like, these COVID vaccines came out so fast, and we're like, it's been in the pipeline for 20 plus years. And so there's been all that research. It just hadn't been rolled out like that before. So you've been listening to Science Coast. The views, thoughts, and opinions expressed are the speaker's own and do not represent the views, thoughts, and opinions of Lewis University. The material and information presented here is for general information purposes only. The Lewis University name and all forms and abbreviations are the property of its owner, and its use does not imply endorsement of or opposition to any specific organization, product, or service. This podcast was produced in the WLRA podcast studios at Lewis University. Visit lewisu.edu for more information about Lewis University. I'm Mallory Havens, and I'm with
Chris White.
And today,
Sarah Powers.
Thanks for listening.
Thanks, everybody. See you next time.